Right. Welcome back for the second part of this introduction to population genetics theory. And so in this part, we're going to talk about the concept of Hardy and Weinberg equilibrium or Hardy-Weinberg equilibrium, which was independently developed by Hardy and Weinberg. So what this is about, and so we're going to first approach that from the perspective of the relationship between alleles and genotypes frequencies, which were introduced in the previous part. So let's now introduce some notations and consider that we have a population, or think about it as a set of individuals, uh, and we have capital N of those individuals. And those individuals are called here diploid because they just like humans, they will have a mom and dad. So right, their genotypes are fully defined by the state of alleles at the state of two alleles. And so at that particular locus that we're going to be interested in, there will be two alleles, and for simplicity, we're going to call them the allele 0 or the allele 1. Right, you can think about 0 being any of the letters that the nucleotides that you want to be interested in, and 1 can be anything. It can be an A, a G, a T, or a C. Right. Again, introducing notations, we're going to call small n 00 as the total number of individuals who are homozygous, uh, who are homozygous at that particular locus with the 0 alleles, right? So they have the 0, 0 genotypes. The 0, 1 or the 1, 0, we're going to not distinguish them. We're going to assume that this is the same genotype. And we're going to call small n underscore 0, 1 as the total number of people with that particular genotype. And finally, the n, small n 1, 1 will be the total number of people with the 1, 1 genotypes. And similarly, we're going to denote small n underscore 0 and small n underscore 1 as the allele counts in the population for how many people have or how many alleles 0 exist in that population, or how many allele 1 exists in that population. Okay, so I guess a simple relationship that is easy to grasp is if you sum all the genotype counts, we have the total sample size, right? Because individuals can either be 0, 0, 0, 1, or 1, 1, and if you sum all of them, well, you have your total sample size. Another thing that is somewhat easy to do is to go from those genotype counts all the way to the allele counts, right? And I'm going to show you how to do this because if you're interested in N0, which is the total number of 0 alleles that exist in the sample, the way you do that is by essentially recognizing that individuals with the 0, 0 genotypes, they have two copies of that 0 allele. So how many are they contributing? They are contributing two of those alleles. So whereas people with the 0, 1 genotype are only contributing one copy of the 0 alleles and symmetrically one copy of the 1 allele. So which means that if you want to count how many 0 alleles there are in that particular population, you essentially calculate it by twice the number of 0, 0 genotypes plus 1 times the number of 0, 1 genotypes. And similarly, how many 1 alleles there are will be calculated as twice the number of 1, 1 genotypes plus 1 times the number of 0, 1 genotypes. So which means now if we are interested in allele frequencies, so what is the frequency of the 0 allele or the 1 allele? Well, again, here we're looking at diploid individuals. So each person has 2 alleles. So how many alleles there are in total in that populations of n individuals? There are actually 2 times n alleles present. And so therefore, to get the frequency, we are simply dividing N0, which is the total number of 0 alleles that are present in the population, divided by the total number of alleles, which is 2 times capital N, and that gives us the allele frequency P0. And similarly, we can calculate this as with the relationship we've seen before, which is essentially the genotype's frequency of the 00 plus half the genotype frequency of the heterozygous. And we could do the same thing for the frequency of the one allele, which will apply. We can follow exactly the same principle, but here we can also use a shortcut because individuals can either have the one allele or the zero allele. So which means that the frequency of the one allele is essentially one minus the frequency of the zero allele. So we can also use that shortcut. Once you've calculated the allele frequencies for the zero allele, you can deduce the frequency of the one allele as one minus the frequency of the zero allele. So what have we learned here? I guess we've introduced some notations, but we've learned that there is a natural relationship that exists between genotypes, frequencies, and allele frequencies, which is that if you know the genotypes frequencies, you can predict the allele frequencies deterministically, right? There is a simple relationship which is shown here. However, what is harder to do, and there is somehow a sort of a mathematical impossibility here, is that if you know the allele frequencies, you cannot predict the genotype frequencies simply because this is an underdetermined problem where you only have two variables and actually only have one variable, which is the allele frequency. And you want to predict two variables, which are your genotypes frequencies as well. And so this in general doesn't have a solution. This system of equation does not have a solution. But what is interesting is that Hardy and Weinberg have shown that under some set of a set of assumptions, we can actually predict relatively well what will be those genotype frequencies from knowing the allele frequencies. And so this is what is called, so there's the set of all those assumptions when they hold is referred to as the Hardy-Weinberg equilibrium. So, and this is the principle. So if genotypes and allele frequencies are constant from one generation to the next, then the population is said to be under Hardy-Weinberg equilibrium. And as we see, for that to apply, for the allele frequencies and genotypes frequency to be constant or near constant from one generation to the next, a lot of things will need to happen. But if that happens, then that Hardy-Weinberg equilibrium holds. And this is what is the consequence of that is going to be shown now. So under this particular assumption, you can predict the genotypes frequencies. So here I've changed the notation a little bit. So just to simplify, we still recognize our 00 genotypes, 01 and 11 genotypes. But for simplicity, just denoted p, small p as the frequency of the one allele, right, which is now shown on the x-axis. And each of the curves now represent the genotypes frequencies. And so the Hardy-Weinberg equilibrium predicts that you can predict the genotypes frequency of the 1, as essentially that p squared, so multiplied by itself. The frequency of the genotypes of the 00 genotypes here can be predicted by 1 minus p, essentially the frequency of the over allele square. So that so far it's very symmetrical. And we can also predict the frequency of the heterozygous by 2p1 minus p, right? And so what I'm showing here, the sort of lighter colors, green, gray, and pink, are what is predicted by theory. And all of the dots are here actually from simulation. So if you perform a simulation with a large enough population, and you can predict that those are the observed genotypes frequencies, and they match fairly well the prediction. And so what I've shown here, going from allele frequency to genotypes frequencies, those equations are shown in the context of diploid species like humans, but you can generalize them to other types of populations that can be polyploid, so they can have more than just two parental chromosomes. And there is also a version of that for sex chromosomes. So here is also assuming that we're looking at autosomal um, chromosomes. So, okay, so what are those assumptions for the Hardy-Weinberg equilibrium to hold? So if we want a constant allele frequency, we have to assume that anything that is creating changes in allele frequency is absent. And so, again, thinking about our population being defined as some sort of collective, we don't want to have any influx whatsoever. So, if you think about migration as a potential source of influx of genetic material into my population, well, you don't want that because that will mean that allele frequency can change as a result. So, one of the assumptions underlying the Hardy-Weinberg equilibrium is that there is no migration happening. Or I guess if I can right away tone down those statements, is that the effect of migration is negligible. Because as you will see in the next part of the next part of this part, you will see that the way we measure Hardy-Weinberg equilibrium is usually using a statistical test, and therefore it's never really strict as we're not looking at the strict version of this equilibrium. We're just looking at seeing whether the data supports those assumptions by and large. So here I'm saying no migration, but just think about it as the effect of migration is negligible. And in the same way, we can think about not having the effect of any mutation, which again is a genetic material, genetic information coming spontaneously in the population. And again, we also have to think about this to have a negligible effect. And other forces like natural selection will also have to be assumed to have a negligible effect. There is another important assumption underlying the equilibrium is that there is random mating, essentially that people are combinations of mates or partners or spouses are largely random. And so we know that it's not true. I think there's a lot of evidence that partners do not choose each other by random. There are a lot of selection criteria going underneath. But as long as it's near random, that could also is one of the conditions for the Hardy-Weinberg equilibrium to hold. What I presented here is the context of diploid individuals undergoing sexual reproduction. And also a final assumption here is that the frequencies of what the allele is the same between the sex groups, right? And the other one is a bit of um, uh, a technical assumption, but we will cover that during the practical, is that the population has to be large. And this is a tapping into an evolutionary force called genetic drift, which we're going to talk about more during the practical. So just that gives you an overview of some of the assumptions that are underlying the Hardy-Weinberg equilibrium. So now let's think about a concrete example. How can we test if Hardy-Weinberg equilibrium holds? And so deviation from the Hardy-Weinberg equilibrium are usually detected using a statistical test. And that test, the test statistic is usually assumed to follow other Hardy-Weinberg equilibrium to follow a chi-square distribution with 1 degree of freedom. And so let's consider the following example where we have three genotype classes, capital A, capital A, capital A, capital B, capital B, capital B. And for each of those genotype classes, we have the counts. So we have 125 AAs, 225 ABs, and 150 BBs, which sum up to a total of 500 individuals. So as we've seen before, what we can do here, we can calculate the allele frequency. We can, as we've seen before, essentially twice the number of AAs plus 1 times the number of ABs, and we divide the whole that whole quantity by the total number of alleles, which is 2 times 500, and that gives us a frequency of 0.475. And we could, as said before, we can use the same principle to calculate the frequencies of B, but here we can also just deduce the frequencies of B as 1 minus 0.457, which gives us 0.525. From this, we can also calculate the genotype's frequencies, right? We have the counts, we have the total, we can deduce the genotype frequencies. And then we're going to use a very important quantity for our test statistic, which is to calculate the expectation, essentially how many of those genotypes do we expect under Hardy-Weinberg equilibrium. So what I've shown you earlier is that under Hardy-Weinberg equilibrium, we can predict the frequency of those genotypes knowing the allele frequencies. So for example, the frequency of the AA genotype should be the frequency of A square, PA square. But here, because we know the total sample size, if we just multiply that PA square by the total sample size, that will give us our expected genotype frequency. And so, sorry, our expected genotype count. So here we would expect 112 under Hardy-Weinberg equilibrium. And so now we can generalize this and calculate those expected genotypes for each of the genotype classes. And the way we calculate the Hardy-Weinberg equilibrium in this context is essentially contrasting the observed genotypes counts with the expected genotype counts across the 3 genotype classes. And here we contrast them with the statistic, which is observed minus expected, the whole thing square, divided by expected, and we sum that. And so here we're getting a test statistic altogether that equals 4.78, which if you were to compare with the quantile of the chi-square distribution of 1 degree of freedom corresponding to a 5% significant threshold, that will be 384. And so here the test statistic is larger than that threshold, which will suggest that we have a deviation, a significant deviation from Hardy-Weinberg equilibrium. So that is the general formula, which I sort of presented, I said orally in the previous slides, and now we can visualize it. So our test statistic is calculated as such, is the sum across all the genotype classes of the observed count minus expected count square in the numerator, divided by the expected count on the denominator. And I've also shown you how to calculate those expected counts. So just wanted to summarize this second part. So a population can be characterized by the frequency distribution of alleles and genotypes. And what we've seen is that under certain assumptions, genotypes frequencies can be predicted from allele frequencies. And I've started by showing that this is usually not possible, but under those assumptions, and which we group, all those assumptions are grouped under this concept of Hardy-Weinberg equilibrium, we can predict those genotype frequencies from allele frequencies. And the Hardy-Weinberg equilibrium can be tested statistically using data. And so this Hardy-Weinberg equilibrium can also be extended to sex-linked loci in polyploid individuals. Here I mostly showed you things in the context of diploid individuals and autosomal variants. But as I said, we can extend that, and that's something you can find in the literature. And finally, deviation from Hardy-Weinberg equilibrium can be used as a way to inform non-random mating. But more generally, if you have a deviation, it sort of suggests that one of those assumptions, if not many of those assumptions, might be violated. And this is something you will cover in the GWAS lectures, for example, as a way for you to detect issues with quality or genotype issues in the data. So I thank you for listening to that second part. It was a bit more, let's say, hands-on in a way, because we talked about formulas and some of the things. But I hope you're ready to embark on the last part of this lecture.